All right, section 2 for chapter 13. We're going to look at the maturation process of both the B cells and the T cells. And specifically for these cells, each of them have what are called their B cell or their T cell receptor. That is critical for the diversity of these cells, which we'll talk about. And each of them have a very specific structure that is important for their function. So if we start with T cells, all right, the T cells, if you remember, they are created within the bone marrow, but they very quickly move to the thymus gland. All right, it's within the thymus gland that it receives, all of those B cells, receive the proper hormones and chemical signals to become and to mature into a properly functioning T cell. Now, along with the T cell receptor itself, you also have other receptor, basically, or sometimes called a co-receptor, that act with the T cell receptor itself to facilitate proper activity. So there's a CD3 receptor. This is another molecule that sticks in the cell surface right next to the T cell receptor, and it's going to be on all T cells. And then you have CD4 receptors and CD8 receptors. Each T cell will have either a CD4 or a CD8 receptor, never both in terms of mature T cells. All right, and those are going to be accessory receptor proteins. So, if we move to the B cells, all right, the B cells are called B cells because they were first discovered in birds inside their bursa, which is kind of similar to the spleen. And if you remember, the B cells are going to mature first in the bone marrow, and the bone marrow has what are called stromal cells. All right, the stromal cells are what gives the B cells the proper chemical signals, the proper hormones for the B cell to actually develop. And then kind of in that naive stage or that immature stage, let's go with teenage years, all right, the B cells are going to move from the bone marrow out to the different lymphatic tissues, specifically the lymph nodes, the spleen, the galt. And if you recall from, actually I'm not sure if we've talked about this or not, but the galt is the gut associated, yes, we did talk about this. This was a quick mention in chapter 12. The galt is the gut associated lymphoid tissue. And so the B cells go there, and it's there that they're going to receive the proper signals. All right, here is figure 13.3. All right, the first part is going to look at the B cell surface. So with the B cells, you have this B cell receptor, right? It's called an immunogen or immunoglobulin, sorry. And when it is attached to the cell surface of the B cell, and you see multiple B cell receptors stuck to the surface, it's called the B cell receptor. All right, included on the B cell are these molecules called MHC class II molecules. MHC class II molecules are going to be found on antigen-presenting cells, which we'll talk about in section 3. The T cell, the right side of figure 13.3, looks at the T cell receptor and its complete complex. So this is more complex than the B cell receptor, but here you have the actual T cell receptor itself. You have those CD3 molecules that sit right next to the T cell receptor. And then you have this CD4 molecule. The CD4 molecule is going to give the name of this cell a CD4 positive T cell or a helper T cell. And if it were a cytotoxic T cell, instead of seeing CD4 here, you would see CD8. And those cells are then called CD8 positive T cells. All right, so T cell receptor, more complex than the B cell receptor. The B cell receptor is actually quite simple. It's just this Y-shaped molecule. All right, this is an important table, table 13.1, because it compares and contrasts B cells and T cells. All right, so you have the site of maturation, bone marrow cells for B cell, or bone marrow for B cells, the thymus for the T cells. The specific cell surface molecules, all right, this is talking about that receptor. All right, the B cells have B cell receptor, also called the immunoglobulin. T cells, T cell receptor, along with other CD molecules, those CD3 and CD4 or CD8. All right, circulation in blood. B cells actually circulate in very low numbers. They are just going to hang out in the lymphoid tissue, the lymph nodes, the spleen. But T cells, on the other hand, are seen in quite high numbers. All right, the receptor for antigens, right, it's that receptor. And then let's go down here, production of antigenic stimulation, or product. All right, so what happens when that specific cell sees its proper antigen? All right, then for the B cells, plasma cells are created and memory cells are created. For T cells, you have different types of activated T cells, also memory cells. So if you remember, the two major components of the third line of defense is specificity and memory. And you see that both here. General functions, all right, production of antibodies for the B cells, and we'll talk about that in, I'm not sure, section 4 or section 5. And then T cells are activated to help other immune cells or to suppress or kill abnormal cells. All right, and unfortunately, they will also mediate hypersensitivity reactions, but we'll talk about that later on. All right, so then if we look at the B cell receptor, all right, it can be called an immunoglobulin. All right, and if it is an immunoglobulin, the immunoglobulin can be attached to the surface of the B cell, and if it's attached to the surface of the B cell, it's the B cell receptor. All right, so an immunoglobulin can be a B cell receptor. If the immunoglobulin is secreted and it's pushed out of the cell to move throughout the body, move throughout the bloodstream, then it's an antibody. All right, so an antibody can also be an immunoglobulin, just like the B cell receptor can be an immunoglobulin, but an antibody is not a B cell receptor. So you have to remember that these two, or that the location of the immunoglobulin gives it its proper specific term. All right, so immunoglobulin structure. All right, we saw that a few slides ago, looking at that Y-shaped structure. And instead of focusing on this, we're going to move to the next slide, and I will read what this says. All right, so if we look down here at the Y-shaped guy, this is the basic structure of an antibody. The antibody is going to have a couple of different structural features that you need to know. All right, the top part of the antibody is called the variable region. The variable region is variable, and so then you can have diversity. You can have lots of different antibodies or B cell receptors that can specifically attach to epitope 1, epitope 10, epitope 1000, epitope 1 billion. The bottom part is what's called the constant region. And the constant region is actually going to be kind of more or less stuck to 5 different types of constant regions. All right, then how do you get this variable region being specific for antigen 1 or antigen 1 billion? Well, that's going to happen at the DNA level. All right, so the DNA level, you've got your DNA here, and you've got all of these different colors, kind of look like piano keys from your kid's xylophone. And each color here represents a section of DNA. And when the DNA goes through the process of transcription, moving to the mRNA component, different sections of the DNA come together to create different color arrangements. Right? The different color arrangements are going to indicate different components of this variable region. It's the variable region where the antigen is going to attach, right? Or where the antibody is going to attach to the antigen. All right. Then you also have this larger piece on 2 sides. So each antibody has 2 heavy chains, and each antibody has 2 light chains. The light chain is the lighter if we talk about weight. And then the 2 heavy chains are going to be connected with disulfide bonds. The light chain is going to be connected to the heavy chain with a disulfide bond. All right, so that is the basic structure. All right, this slide shows 2 terms that you need to know, and both will apply to B cells and T cells. So the first one is clonal selection, and that is, as you see here, the mechanism by which the exactly correct B or T cell is activated by the antigen. And then clonal expansion is the rapid multiplication or expansion, increase in number, of that specific B or T cell after being activated by the antigen. And if we look at the slide here, again, this can apply to either a B cell or a T cell. And what you have here are different either B cells or T cells. All right. And what you see here are the specific types of receptors, and they are all given just a slightly different shape or a different color just to show specificity. And so this guy is going to actually react to a self-antigen. All right, you don't want your immune cells reacting against your own antigens, your own cells. Otherwise, then you develop autoimmune disorders. All right, same with this guy. All right, there's a specific interaction between the receptor on this cell and this self-antigen. So then, if that happens, this cell knows to program itself to die, and so that's why you have the X. And what are left is this repertoire, this group of B or T cells that have a very specific receptor. And then in this case, all right, this epitope actually comes to and binds to this proper receptor. All right, and that is meaning, or that's the clonal selection, where this one clone, this one cell, is going to be selected because it has the proper antigen, and then it replicates. All right, and that's the clonal expansion. And it's clonal because it's a clone. It's the same. All right, so real fast, concept check here. For each of these properties, determine if it describes a T cell or a B cell. All right, so matures in the bone marrow. B cell, you've got B and bone. High numbers of circulating cells in the blood. All right, if you refer back to that table that compares and contrasts B and T cells, you'd remember that it's T cells. Requires antigen to be presented with an MHC molecule. That's going to be a T cell. Produces antibodies. All right, antibodies is the main player. That's an immunoglobulin within the B cells. So B cells for option D. and cells are activated to help other immune cells or to suppress or kill abnormal cells, and that's going to be a property of T cells.